Hello, good afternoon, good morning to some of you. This is Harold Bilt from NIZO Food Research. We'll be now starting the webinar on sensory evaluation of foods, the advanced techniques, which is a follow-up of last May. Welcome all to you, especially for the people from Asia. I think there are some people listening in the evening right now, so also thank you very much for your attendance. Today we will be presenting two of us. My name is Harold Bilt and I'm a senior scientist at NIZO Food Research. And Peter de Kock is also here and he is principal scientist Flavour, also in the same department of NIZO Food Research. In the announcement you've seen that Kerstin Busek would be presenting, and unfortunately she could not today, so I will be taking her place. I'm with the same background, so that should not make any difference. For your convenience, we have actually put pictures of ourselves in the right upper corner, so you can tell from the picture also who's talking. So occasionally we will switch. That's for my part, and I would like to invite now Peter to take over. My slide does not continue, okay. And introduce NIZO Food Research to you. So here's Peter de Kock.
Good morning, this is Peter de Kock speaking from NIZO Food Research. Thank you for joining this webinar. By the way, if you have any questions, you're going to find a chat box in your web browser. So if you have any questions, please type them in. Some of the questions which are of general interest, we will actually integrate in our webinar, and all the other questions will be answered offline or in a private chat box. Back to you. So thank you very much in joining this webinar again. I'll have prepared a very short introduction of what NIZO Food Research is, as probably many of you are not familiar with our institute, but I'm going to make it very quick, and you can have access to the file anyway, to the recording. So you can look back for all the information that we provide you. So essentially, NIZO Food Research is an independent private contract research company in the food industry. We typically do innovation research programs with a very strong scientific background in making innovations possible in the food product market to help our clients, enabling bringing innovation to the marketplace. So most particularly those problems that are technically too advanced or are outside your core competencies, you could always try to contact us and see whether we can help you with that. We are located in the center of the Netherlands, close to the University of Wageningen, which is called the Food Valley area in the Netherlands, and we have offices in France, in the UK, in the US, and in Japan. The whole institute is about 180 professionals. Here on this slide you can see our central research facilities, including this building, and this one is our food grade pilot plant. So we have a significant pilot plant that enables us not only to actually produce foods and food model systems or ingredients on small scale, but also take it up to pilot plant scale. So essentially what we do is we look into research areas that cover food safety, that cover flavor, texture, health and processing, and build them into applications. We have an open access research network. We do quite a bit of consultancy and product process development, and actually producing test products for instance, test markets. Our main target is working to deliver together. This is a very strong motto of us. We could never do this without you, and we hope we can help you in assessing or actually achieving innovations, and that's in terms of new processes, new formulations, in test productions, as I already mentioned, but also I would say medium to small scale toll operations, and that we actually do in our pilot plant. Our pilot plant has a motto, actually, business as unusual, because the usual business is probably something you can do yourself, but making something happen on a relatively large or small scale, or making test production runs, is something which probably requires a well-facilitated pilot plant. But you may have joined, I'm not sure whether you've joined also our previous seminar, which was held on June the 20th this year. This was all about basic techniques in sensory. And some of the conclusions from that session was that indeed you need sensory testing to connect, I would say, the technical specifications of your product and connect it to consumer liking. This is, I would say, really a link that is required for making that kind of transition. For sensory purposes, you have different methods, and it's very important that you apply the correct method or the most applicable or the best method for generating your sensory data, enable to say something about your the perception, the sensory perception of your food, and also being able to do this connection to consumer liking. Every panel needs to be a dedicated panel, in that terms that they need to be, I would say, well trained on the specific task that they have, and that includes the way you ask, actually what you ask to the panelists. And this is pretty important, that you propose them the exactly right question, and that they are being well equipped for dealing with this task. And quite important for us is also that you consider that even panelists, even they are well trained, they are not a real analytical instrument, but they are still humans. And that will actually also be a subject of today. What you're going to see is we're going to talk a bit on cross-model effects, which is in fact an interaction of many kind of sensory percepts that are being integrated in the human being, and that's, I would say, a psychological process. But also for panel motivation, it is very important that you treat them well, so that they service you well as well. Now, the content of this webinar is actually, we're going to look very shortly in a very concise presentation on olfactometry. I'm going to explain what this technique actually stands for, what it is. The second thing is we're going to address release and temporal effects of aroma and tastings. What we're also going to do is show you how we actually build systems to, I would say, prepare a virtual food in terms of its taste and aroma sensation. And as I already introduced, we're going to look at cross-model effects and how these affect mood and the judgments, the sensory judgments of foods. Now let's start with the first one. Olfactometry is in principle an analytical sensory technique which tries to establish which aroma compounds in formulations are really relevant and contribute to the overall aroma perception of an aroma and a taste. What we essentially do in this technique is you take an extract, or you take actually the aroma system or taste system of a food, you make a set of dilutions from a strong dilution to a weak dilution, and you submit all these dilutions to a GC. Now, a normal GC sends all the information to its analytical detector in producing your GC chromatogram, but what we do here is we actually split this in two just before it reaches the analytical detector. One actually still goes to the analytical detector, and the second stream goes to your human assessor. This is typically what you see happening here. This human being is being used as a detector for all the sensory output that comes from the column. So in this particular case, she is being asked to identify odors that actually elute from the column while the chromatogram is being produced. What then happens, in fact, if you most particularly do this with a panel, because all panelists can be selective in their sensitivity towards a set of compounds. What then happens is that people respond to an aroma that elutes from the column. Some compounds will actually survive the dilution steps, but some compounds will drop below that threshold. This one, after one dilution, already is gone. The second example, it requires 2 dilutions before it drops below its threshold. And then theory says that the compound that survives the most dilution steps contributes the most to the sensory perception of the overall aroma. So the rationality is the stronger the dilution it takes to actually remove the sensory aspect of that compound in the complete cocktail is proportional to the importance of that particular compound. Now, one of the outputs is of course from the analytical trace, the regular GC. And it's very easy in this particular example to identify more than 700 compounds which are all present in coffee. But the realization is that not all these compounds are sensorically active. Now, this technique actually shows you which of those compounds have a sensory effect and which are just happen to be present but do not significantly contribute to the overall sensory perception. And we marked marked these compounds in red. So all the red dots represent compounds that do contribute to the sensory percept of the coffee aroma. And this amounts to about 30 compounds that actually do the trick. Now, some of these compounds are high, which means that they are relatively high in their concentration. So if you would integrate the peak area or even would quantify them, you're going to see that it requires a significant amount of this compound to generate an effect. Some of them, for instance, this one has a high peak, but is sensorically very much less interesting, this one as well. And in some cases, like the one you see here, you might see a small peak here. If you would zoom in, you couldn't even see a peak of this particular compound. And this means that this is a highly potent aroma chemical. And that's the power of this technique. It doesn't really show you the analytical trace, but it shows you what is called an aromagram. Now, the next job is, of course, to identify this whole set of compounds, and that you can take to actually build new aroma sensations, or to look into the quality of the aroma depending on what happens in your product. And for the latter, I brought an example as well. So what we did is we took only the aroma compounds which are sensorically active, and we used them to put the response of these of the peak areas. We've been putting them in what is called the PECA pots, and that actually we have been covering in the previous webinar as well. So what we there used for evaluating sensory results, we now also use for analytical results as well. If you would plot all the compounds, the chance is very big that you would get rubbish out. But if you only focus on those compounds that indeed have an active role in the sensory perception of this product, then a picture starts to emerge that makes sense. So in this particular case, in coffee, you see the 3 different species in coffee. This one, which is a Robusta, which is an Arabica-based coffee, and this is an instant coffee. And next to that, we plotted the difference between fresh and old coffee. So this enables you not only to discriminate between different types of coffee, but it also shows you the effect, the chemical effect, I would say, from aging of the coffee. And that's a very powerful method. So on one hand, you get consumer information or physical data or, in fact, sensory information. And now it starts to make sense in terms of its chemical composition. As I indicated already, this is a very small introduction. There's lots to be said about this technique, but we're going to progress with our aroma release. Because there is a realization as well that not only the aroma composition is quite important for the sensory quality of your product. Next to the aroma being sitting in a product, it needs to come out during the mastication of a product. And that's typically what we call aroma release. Now, mastication in itself is a very complex system. It starts already by assessing a product before you even start masticating. So before the first bite in the mouth, you already make a kind of assessment based on your sight, the smell you see, the way it is being handled in your hands, and in fact whether something is being packed in a crispy packaging. Everything already builds you a kind of conceptual idea of the quality of this foodstuff. Then you put it in your mouth, and then lots of things happen. You're going to cut it with your teeth, you're going to add saliva, probably the temperature changes, and that is a regime which changes over time. And then you swallow and actually do the rest of the perception, both in terms of flavor and texture. This whole process is very dynamic. Most of the aromas that are being exposed, to which you are being exposed, or taste compounds, they have a kind of time regime. So if you plot intensity versus time, you always get these kind of curves. An aroma perception goes up and goes down. It can go up fast and high, or slow and much more prolonged. So this one is much more strong, I would say, in aftertaste. But it shows that perception indeed is dynamic. And that depends on quite a few parameters. For instance, the ingredients are pretty important. Whether it's high in oil and fats, proteins, or hydrocolloids, or emulsifiers, has a tremendous effect on how this curve is being plotted in terms of intensity versus time. The texture is, of course, very important. We'll come back with an example in textures that are different, I would say, in firmness, in viscosity, if you like, and that has a completely different effect on how the flavor comes to expression. Of course, the flavor properties are important, whether the flavor compound is lipophilic or hydrophilic, whether it's highly volatile or less volatile. But also, I would say, person-to-person conditions, like the geometry of your oral cavity, the behavior during mastication, as well as the amount of saliva you produce during the eating process are pretty important parameters. All this is what we call the aroma release, and we have a technology for measuring this. What we actually do is we take a person who is eating a foodstuff, we take small aliquots of air from the nose space, as close as possible to the olfactory epithelium, where the sensory perception is actually taking place, and we run this through an MS which works at atmospheric pressure for the convenience of the panelist. What you can then do is actually make a concentration profile of the aroma volatiles that are being accumulated or that actually are being released into the nasal cavity. You can do this when people are sniffing a product, which we call nasal perception, or you can also do this when people eat a foodstuff, and this is typically called retronasal perception, because it's retronasally that the aroma is being released from the foodstuff, and while the product is being swallowed via the inhalation-exhalation cycle, is being introduced into your nose space for perception. This, as I mentioned already, we do with a MS-based system. The MS you see here is an ABCI MS. It's taking again small aliquots of air out of the nose pocket of a person who is actually drinking or masticating the food. And the convenience of this method is as well that you can ask these people to rate their time intensity during the course of the eating process. So on one hand you will receive analytical data, and on the other hand you get time intensity information as well. And Harold will go into the details of time intensity measurements. This is, as I mentioned, this time intensity sensory information coming out of this, as well as the analytical information that comes out of this. And this is much easily accessible for making correlations. This is a typical example what you see if you attach somebody to an MS breath. This is not noise actually what you see. This is the inhalation-exhalation cycle. And when people inhale, there's virgin air coming from the outer room, and immediately the system says, I do not detect any aroma compounds. Then somebody exhales, and then the food is actually being brought in contact with your air space in your nasal cavity, and the signal goes up. Now this curve is rather, I would say, noisy in itself. And what happens in your brain, which is a special algorithm, is that it's being curve flattened in some kind of curves that you're probably familiar with. Intensity goes up and goes down. And again, these analytical data, they can be transferred or matched with sensory perception. Another example shows you what the effect of aroma is in terms of low and high fat product. So typically, if you take yourself a product and remove the amount of fat, what you'll see is that in a full fat product, in most of the time, actually the aroma release is lower compared to a low fat product. That's just a way of flavor being partitioned between the fat. And in this particular instance, a hydrophobic aroma is being readily dissolved in the fat, so its headspace concentration actually drops. But I would say that's easily correctable, because the only thing you have to do is add more aroma to this particular product. But what you then discover is that it doesn't match at all. What you see that the full fat has a much more prolonged aroma release than the low fat product. Now this might not be significant, but if you just consider a certain level of intensity in terms of its aroma release, it is quite critical. You see a significant introduction of what we call aftertaste in this particular case, and even we're going to show you a bit further down this webinar that people actually use this to make a kind of hedonic quality rating of the quality of your product, as well as the calorie intake they have consumed, they've been generating during the consumption of a food. This means that not only the intensity is quite an important parameter, but much more also the duration of the aroma along which is being released during the mastication of a foodstuff. Now, this, of course, requires quite a bit of technology to implement in your food. That means also that you want to test these kind of constructs before you actually start making a product. And that's exactly why, and we'll come back to this later as well, why we generated a platform where we can mimic these conditions before you actually prepare yourself or start developing yourself a new product. So the next thing is about time intensity profile, as I already indicated that we would cover. And this actually will transfer the webinar back to our adult, who is going to take you through a series of slides involving time intensity measurements. So Harold, the floor is yours again.
Hello again. We'll be continuing with some issues on time intensity measurements. Time intensity measurements are, in a way, not very extremely different from what has been explained last time on QDA paneling. You will still be using a panel leader and similar kinds of panelists or consumers as you will be using also for QDA, especially the panelists normally would be the same actually. What you do actually, you will focus only on one attribute. So with QDA, you eat the food, and after you've eaten the food, you will rate a number of intensities on attributes. In this case, in time intensity, you will focus on one of those attributes at a time. And first, of course, these attributes have to be explained to the panelist, and then a sample is given, and the panelist measures the intensity of this attribute during time. So you will actually be holding a mouse, or you will maybe even holding something else that you can use to indicate what intensity you have sensed at that moment in time. And they are being sampled, those responses, and recorded by a computer. And when the person is done eating, that will also be the end of the measurement of that specific attribute. If you would like to have more intensities of different attributes, then you will have to repeat that sequence, of course. And again, also, if you would like to do it for the whole panel, then you will have to repeat it over panelists, which makes time intensity measurements in the general actually a more time intense or more laborsome technique. If you have done those experiments, if you have done those measurements, then the curve of the intensity can be plotted, and that's as a function of one attribute over time. And this is an example of that. You see here 3 curves that represent time intensities, and this is to one odor example, and that's the ethyl heptanoate, a fruity kind of odor component that is released over time. And people have done this in this case for 3 different products in which the oil contents are different. And as you remember, that Peter just explained to you that if you have different oil contents, it can actually be that the odor component is either delayed in its release or it's actually captured by the oil, and in that case it will be released less, or the opposite takes place, that the relative concentration of the component, if it is hydrophilic, then it will be higher in the water phase and it will be released more. So in this case, what you see here is exactly that. The person releases, rates the intensity of the odor component over time, and the blue curve represents the high oil content, and you see that over time there's a general increase of its intensity. In this case, you see that the profiles are rather the same. It does not necessarily have to be the case, of course, as you saw in the parts on the flavor release that Peter presented. This is a nice technique to see actually not only what are the intensities, but also how do they develop over time. If you go a little bit deeper into that, and I will not be talking too much about this, but just for you to understand that you can do a lot with these results that you couldn't do with QDA results. People in industry have been looking at these time intensity curves and have tried to extract a number of parameters from these that indicate something on how people perceive, or that may indicate on something how people perceive in general these products. And in this example, you will see that, for instance, you can look at the ramp, so the gradient in the beginning of the perception where people increase their intensities, or you can look at the decay over time, which is, as you saw before, often a function of oil content. And a number of other things that you can do. You can take partial areas of the curve, you can take the total area of the curve, or only the area during the latter part of the decay. I'm very dreadfully sorry for what just happened. We had a system crash here, which of course normally never happens, only when you're online. So sorry for that. I've shown you the graph, the different parameters that you can extract from the time intensity curve, which is the previous slide to this one. I don't dare to show it to you again because it might be the slide that caused the trouble. But what actually you see here in this table is that a number of these parameters that you can extract, like the ramp, or when it's first time peaking, or so when it's the first maximal value in time, and a number of these, or what's the altitude of the plateau. All these parameters you can actually test and see whether they are sensitive in their value to perceive differences between the different products. And what you see in this case, depending on the product actually, and depending on factors like oil content, there are different parameters relevant for perceiving that difference. And in this case, for instance, half the area of the full curve, if you have a high fat content, I think it is in this case, if you have a high fat content, then you see that that parameter responds to that significantly in differences. Not all of them do, and unfortunately, depending on the product category, you will need one or the other. The conclusion that you should draw from this is that time intensity curves themselves can relay a lot of information about how people perceive and how they perceive differences. But for every product category, you have to be really critical on what information you extract from these curves to be able to distinguish. And of course, people, when they perceive, do this automatically. And in our case, we have to do extensive studying on these curves before we know which part of it is the relevant part. And it was indeed fat content that caused these differences. From time intensity, I would like to go now to another technique that is somewhat similar to it, that is called temporal dominance of sensation. We abbreviate that to TDS. It started already 10 years ago with Pascal Schlich in France, and it's now starting to be picked up by the community, and we are using it also extensively nowadays. It is similar in the way that you ask people to do something continuously over time. You do this in panels again, and you use trained panelists as you would use for QDA testing, but they have to be specifically trained also on the task. What you do use is a list of attributes like you have from QDA, and typically those attribute lists are somewhat smaller, so instead of having a list of 10 to 12 or 15 sometimes attributes, you use only 5, sometimes 6. And the reason for that is that people actually will continuously indicate which one of those is for them dominant. So they're very much trained in doing this with 5 attributes at a time, and when they are given a sample, they immediately choose which of those 5 attributes is dominant. And that can change over time. So if you perceive sweetness, for instance, after a while it might become more slimy. So then the switch that you make is from rating that sweetness is the most dominant to sliminess being the most dominant. And this is done always in an immediate case, so if people perceive the difference, they should make that rating. It's quite challenging for people sometimes, so that's why they have to train. That is done during the full bite, so when people finish eating and they have cleared their mouth and they say they're done and they don't perceive any aftertaste, that's the end of that rating procedure. And they will indicate then also that they are finished with eating. And from all those panelists, the data will be gathered together, and that produces something that you call a TDS plot for each attribute. You have then a specific curve. And if you have a list of attributes like these here that are chewy, slippery, sticky, smooth, liquid, and the last one saying I'm done, you can do this, for instance, for gummy bears, which are chewy candies that you have to chew for a while before you can swallow them, which means you need some time to evaluate them, and during that time texture changes and taste changes as well, and people can reflect that change in what they do with the TDS. So then this could be an outcome of that full panel response. You have to imagine that these are accumulated responses, so this means that 0.8 of all the people in the panel were actually at this point in time saying that something was chewy. And after a while you see that some people actually start responding more on stickiness, and therefore also decrease the total panel response to chewiness, and at some time sticky will become more dominant than chewy. So you can say that here at this point in time, the general impression of the panel is that the product has become more sticky than chewy. And you can continue this in time again, and after a while the chewy and the stickiness have disappeared, and the product becomes more liquid to the person, to the people in the panel. This is a typical TDS curve, and normally you would be interested in which of the attributes is dominant at which point in time. But the more important thing is also that the significance of these responses should be considered. And you can imagine if you have, in this case, 5 different attributes, then purely by chance a person could be rating chewiness at any point in time, and the chance level is 0.2. So somewhere about the level of 0.2, there is a level where you could say that it becomes significant. You do that with binomial testing, and in the end the significance level is the critical value. So everything above this significance level would be considered a significant TDS attribute at that point in time, and that's how it works. I've said something here on the fact that the scale is normalized, so you do not count the seconds anymore. Normally you could plot seconds here, but what we often do is that this is representing the starting of the evaluation and this is the ending. So if people have different times that they use to evaluate, you can compress or stretch each evaluation and therefore have the different stages of the processing also coinciding in this graph. That's a typical way to do this. And to make it even easier, instead of just evaluating these aspects of the plot here, you can inspect these Gantt charts that you see above the plot, and they indicate the significant regions. The nice thing is that if you have only these, then you can easily compare between products. You can easily plot these for different products and see whether some products have a longer period in which they are chewy, or maybe a shorter period in which they are liquid, and you can optimize your products accordingly. So if you know that for your product it is not really appreciated that it is chewy, then this would be a nice way to reduce the time during which it is dominant in chewiness. So we go to the next part here now, which is instrumental delivery of aroma and taste. Of course, every product delivers aroma and taste, but you can also deliver these separately and mimic the food. So what you could call then building a virtual food. You could now say, why would you like to do that? You can just offer the food, but I will come back to that later. There are reasons to do this actually. I'll give you one example to introduce, and this is on cross-modal aspects of perception. This is a study done by Davidson already a while ago in England. What you see here is that you have something that is called menthone, which is giving you a menthol aroma that, for instance, you often perceive in chewing gums. And these chewing gums have also sugar in it. So these are chewing gums that have a specific time intensity curve for the mint intensity, and you see that that mint intensity decreases initially and then decreases again over time. This is what you would expect. What actually is the case, if you look not at the responses but only on the presence of the menthone, then this is not really in line with what you see for what people perceive. So there's something strange going on. The perception does not reflect the intensity, the concentration of the menthone. If you look a little bit deeper into it, then what you see is that the third curve that is plotted here is representing the amount of sucrose in your mouth during the eating. So this is the concentration of sucrose that is emanating from the chewing gum, and surprisingly the sucrose intensity curve or the concentration curve predicts very well the mint intensity, which is a strange result. So the perceived mint flavor follows the sucrose rather than the menthone release. And why would that be? Because menthone is something completely different than sucrose. The explanation of this is that you have an interaction between taste and aroma compounds. So whereas people think that they perceive mint, what they're actually responding to is sucrose, and it is because both of them are very complementary. Mint is something you smell in often very sweet, tasty chewing gums, and the sweetness itself actually is therefore become a representative of the mintiness, and once it has disappeared, people just filling the mintiness, although it's not there anymore, as long as the sucrose is there. So if the sucrose is gone, then everything is gone. That's what the typical perceptual interaction. And you can use it often in also optimizing your food. Another perceptual interaction that I would like to discuss here is something that all of you may have experienced at some point in time, that if you add a thickener to a product and you try to rate the taste or the flavor intensity of the product, then you'll see that the more thickener you use, the more weak actually that flavor impression will become. I have here the curves of the concentrations of a aroma component that is actually detected in the nose space. That's the technique that Peter showed you before. And this is measured for different gels, and those gels are not the same. They are actually different in their concentrations and therefore produce different hardnesses of the product. If you would look only at those release curves of components, you would say that there are no differences between the gels. If there are any differences, they are not large, they are minor, and the error bars will probably just actually blur all these curves into one. However, if you would ask people now to rate the intensity of the flavor, so you will ask people to use the time intensity curves and say how intense the flavor is over time, then surprisingly, if you look at those curves for gel 1, which is the soft gel, you see that the intensity is perceived as much more intense than the intensity for the harder gels. So although the release curves are the same, the intensity flavor intensity is not, and they actually depend entirely on the hardness of the gel. So the gel hardness here predicts the perception, although it does not affect the release itself, which is again therefore a perceptual interaction. It's a very large effect. It's something that is bothering people in food industry a lot, because if you would like to have a very mouth-filling, a nice, pleasant, creamy, thick dessert, then you will know that you have to use more sucrose, for instance, to sweeten it than if it would be a drink. And that's caused by this effect. And that interaction is taking place at the perception level. That has been shown by Koen Wil here once at the Institute, because the release curves are the same. So in the nose, nothing different should happen. And the last aroma release related study that we performed a while ago is something on systems that use casein as a thickener, and those systems, they have aroma components in them, and those aroma components were selected because in cheese they were known to affect the creaminess perception of the product. We thought that if the aroma component could do something with a textural aspect like creaminess, then maybe we can also use them to change textural aspects of a model product, and that's what we tried here. So what you see here are different casein concentrations. The more casein, the more firm the product is. This is firm, this is not very firm. But what you also see is that over these three categories you have increasing aroma concentrations. What you see there is that those aroma concentrations reduce the firmness again. Structurally there's no difference. If you do TPA evaluations of these products, then you would see that the texture actually is the same. So the aroma component is actually the cause of the reduction of these firmnesses. Again, a perceptual interaction. So from there I would like to go to something that relates to texture perception in your mouth, but it's an entirely different technique. It's very different from what we've been discussing up to now, so I will actually explain a little bit on what the technique does. The technique itself is used to evaluate what a person actually does when he eats. So when a person is disintegrating food in the mouth, we would like to know actually what does the person do with the tongue, how does the person chew, and we do this in a machine that is called an articulograph. And those machines, articulagraphs, are actually made for speech research, and you use them to stick this kind of sensors on the tongue or on the jaw, on the head, you can put them anywhere, and have these actually detect where the tissue goes over time. So if you have these three sensors on your mouth, these sensors can actually then, in this environment, pick up their location, and therefore you can evaluate the movement of the tongue over time. And they are developed for speech research, which means we had to adjust the system a bit to do also eating research, which often means that for the more solid products we leave this location out because there's a lot of interaction in this part of the tongue and we follow only movement on the sides, but it's still possible. You can still use it for eating research as well. And what is more important is that if you have all these locations over time, that still doesn't tell you much really about the eating behavior in itself, because you have to extract meaningful behaviors from it. So you would like to know, what does the tongue do with respect to the palate, for instance. Having said that, if you would like to know what the tongue does with respect to the palate, you actually need to know where the palate is. We don't stick sensors on the palate. We do something instead, and this is a nifty technique to use the same system to get to an individualized representation of the palate of each person. So this is a model head, but in that model head we project a real palate, and this is a more detailed view of the palate. So this is the palate from behind, this is the low part, and you could tell that inside the mouth it would be the lining of the palate that you would feel if you move your tongue against it. And the technique is simple. You use a sensor with a stick on it, and you just touch the palate in a kind of iterative way, and you just follow it until you detect all locations on the palate, and these you use then to construct the palate from with your software. Once you have the palate, you are of course curious on what a person does during a task. I'll give you an example for that. This is some work that we did with caramels in collaboration with Mars. Some of this work I also presented last April. It was in the US on ACS conference, so this is now public. We had 6 panelists, and they have eaten a number of caramels 7 times for each of the caramels, and we used 2 sensors. Like I said, only the ones that are on the lateral sides of the tongue, not the one in the middle. And we used 2 different categories of caramels: the regular ones, the ones that you eat, and low-fat caramels. Those could be a alternative, a more healthy alternative for them. And we used 3 different eating instructions. So we had people evaluate these caramels while measuring locations on the tongue, and we just asked them to eat, or we asked them to eat while performing the TDS task that I discussed before, or to eat while focusing on only a single textural property. And why would we do that? Well, if you just eat uninstructed, it might be that people just do different things than when they are focusing on the sensory aspects of the product. In this case, this category and this category represent very much what people are doing in the laboratory. And this is maybe just what people do when they eat a caramel at home. And we were interested whether these 2 different instructions would cause differences in the way that people eat. The measure that we use is, in this case, the velocity of the tongue. You can plot that for any direction, or you can ask the system to plot it only for directions with respect to the palate, perpendicularly or in parallel. And for this specific velocity measure, you see that there is a clear effect of the task instruction. So people do different things in terms of velocity, and what they look like is that the amount of attention that you invest in actually what you perceive in your mouth is represented here in the speed of the tongue during the task. This is time beginning to end, and an average speed of 1.5 millimeters per second here is achieved, which is not very high, but it means that on average, of course, people sometimes do nothing. But then if you have an attendant situation, then you see suddenly that speeds go up, and that's over full range, especially in the early stages, but you'll still see it in the later stages of eating. So this difference is only caused by instructing the people to perceive what they have in the mouth. This has quite severe implications also, because what you can tell is that if people do much more with the tongue, especially in early stages, they have a double velocity almost. It means that you interact more with the product in your mouth, and therefore the product disintegrates in a different way, and most probably also faster. So the task instruction itself has an effect on the quality of the product in your mouth. And you can expect that this also has implications for your perception for later attribute aspects of that product. So what we do in the laboratory, therefore, does not necessarily represent the product that you actually eat when you're at home. And this is one of the important conclusions from this study. This is for you just to have an illustration of what you can do with this technique. technique is new, it's being developed still, and actually we hope we will be publishing a lot on this in the coming year. So have a look out on publications on the articulography results for this. The next slides actually will be presented by Peter again. So you will be hearing another voice again. Peter will take over for the work that we do on integrating different aspects like texture, taste, touch, sound into one perception, and use that to build a virtual product instead of a real product before you actually can do anything in the lab with the product itself. Here's Peter.
Thank you, Harold, for this introduction. Indeed, as Harold showed, it's quite an intricate kind of concept where lots of sensory interactions are being displayed into, I would say, the sensory perception that people have when they masticate the food stuff. That's what we've seen up to now. That also makes it very difficult from a conceptual point of view as well to develop foods if you cannot grasp how strong these effects are. And this is exactly the reason why we started to build a platform a few years ago already, where we try to integrate all these sensations separately from each other. So what we actually do is we use ourselves an olfactometer to deliver aroma, and we use ourselves a gustometer in delivering taste. So essentially what an olfactometer is it's an instrument where the aroma is being volatilized and being fed into an assessor using a tube in its nose. And by settings of a computer you can actually decide which aroma is being forwarded to the assessor, but also its concentration. And for us, very, very importantly, also the way it's being perceived during time. And so we can mimic these aroma release curves by actually changing the settings in the olfactometer and submit any kind of aroma profile, I would say, for instance, of a low fat, but also of a high fat product, and have the panelist experience what the sensory outcome would be. And again, you can do exactly the same with taste. So our gustometer is in fact a set of computerized pumps. Again, we took this largely also from the information that we got from the group of Andy Taylor, who started with using this kind of equipment. That enables us to have a tailored and designed taste release profile being generated by the gustometer, and that's being provided to a person for assessment. And while he or she is actually is doing this and actually assessing the assessment, we can also ask people to eat the different kinds of foods. So you can change the texture of your foodstuff, and you can change the aroma release pattern, you can change the taste release pattern, and make all kinds of combinations of these different kinds of sensations. And then make yourself kind of virtual food, and test all these conditions depending on the types of release, depending on the type of texture, and how this comes to perception. And perception can be twofold. You can just ask these people to rate their time intensity, or in fact you can use consumers in this platform as well to rate that in terms of liking. But you can also use, as you can see here in this picture, use an EEG response. And the EEG response is scientifically extremely interesting, but for instance, to be able to translate EEG results into liking scores, that is still requires quite a bit of research. And that's something that we will not cover in this. Harold will refer back to it at the final end of this, at the end of this presentation. Now, having this platform being built using an olfactometer and using the gustometer, and still rely on the sensory perception of a person, also enables you to look at integrations of aroma and taste effects. So I only brought one example where we actually use this kind of technology, which is being presented in this slide. So essentially what we do is we screen, for instance in this case, for non-volatiles in the combinations, in the sensory combinations of volatile compounds, I would say in an automated way. Central is again the human perception. So we have a set of panelists that are accustomed to assess these, to use and assess the sensory output of this technique, and we hook them up again to our gustometer in feeding a taste intensity, while actually the aroma is now not being fed from the olfactometer, but from the elution pattern of a GC. And remember that we've covered this GCO at the very early start of this presentation. We use a similar kind of technique in that a GC makes an elution pattern of the aroma volatile compounds, and at our will we combine a set of those peaks with actually the stimulus from a gustometer. Now, in normal cases, as I showed you, a GC would produce a continuous stream of aroma chemicals. That would be detrimental to this technique. So we build ourselves a special GC which collects all the aroma chemicals during a certain period of time, then are suddenly released, and when they are released, we can either yes or no combine it with a stimulus from the gustometer. And then again, as I mentioned, we can ask these people on how they feel, what kind of sensation they experience from the integration, from the aroma and the taste. We do this, I would say, in a well-structured kind of setup. In most cases, you generate first an odorless pulse with a taste pulse, then you have a water rinse, which is all being performed by the gustometer, and then at a certain instant you combine the aroma pulse with the taste pulse. And comparing those two situations actually gives you information on how people integrate the taste effect and the aroma effect of any kind of package that is eluting from the GC. Now, as I mentioned, we can use this GC, of course, but there's many examples in where we use our gustometer in combination with our olfactometer to do aroma delivery, for instance, from all kinds of fruit and spices and herbs types of product. That's one example that we did for a company which is called Vion. Vion is the second largest meat producer in Europe, and they actually were looking at on how to boost the salty impact of their products without actually using salt, of course, but also any kind of taste enhancer set of compounds. So what we did for them is we took a meat extract, a bacon extract, we submitted to the GC in delivering us a whole series of packages of aroma compounds that are present in the bacon aroma, and we combined it with a broth. A broth being, I would say, a model system for any kind of food product, for any kind of meat product they are producing. And the question is, which of these aroma fraction actually has an effect on the salt perception? So you can ask these people to specifically rate a certain sensory attribute. And the result is being depicted on this plot. What you see here is in fact a whole series of packages. So every peak is not a single aroma chemical, but now represents a package of aroma that elute from the GC from this moment until that moment. These are being concentrated at the end of the peak and have been combined with the, I would say, the stimulus from the gustometer. And then we ask these people, please rate, and it's always a panel, of course, please rate whether you perceive some kind of intensity perception. The result is actually that we found ourselves 3 peaks which have a significant effect, and remember these are aroma compounds because they elute from the GC. At least 3 peaks which in combination with the broth from the gustometer have a significant effect in increasing this particular case on salt perception. This means that 3 out of 31 fractions indeed showed a significant salt enhancement being tested by the panel of 10. Everything has been tested in duplicate. And most particularly this peak, for instance, has a 70% salt intensity enhancement. And this can be used, of course, in a salt reduction program. Also quite interesting is the peak at the very end of the chromatogram, because there's hardly any concentration being present in the extract, but it still is pretty potent in terms of its salt enhancement properties. Now this is as far as we can communicate with you the result of this study. You can imagine that the next thing that you do is look into the identification of the compounds sitting in this peak. And the idea was, that we have been doing for Vion, is actually use these compounds to select any kind of raw material that could be used as a salt system. And then of course you can use the raw materials and select them based on any kind of preference they have in terms of those raw materials that fit very well in the pellets they actually want to use in their food system. And so this is a very powerful technique that is being built based on combining all kinds of instrumental delivery of taste and aroma compounds, but still relying on the sensory assessments that people have, because this is the way, in fact the only way in assessing the multimodal effects of foods. Talking about the multimodal effects in foods, most particularly also in combination with mood, This brings us a very, I would say, interesting technology as well. One of the questions we raised ourselves is, do people actually measure the calorie content of a food? Have they some kind of conceptual idea of how many calories there are in a food? And what people seem to understand is that most of the firm foods, solid foods, they judge them to be more caloric intense than most of the liquid foods. Now, of course, that doesn't have to be true, but that's the way people actually assess such a food. They have the, as I already explained, the conceptual idea that there are more calories in a solid food. Now, that's an interesting theory, of course, and the question is, of course, can you make this into applications? So we did a study, in fact, where we used our olfactometer again to deliver the aroma of a product independently, I would say, from the product itself. So we took ourselves a dairy drink. We colored it pink to suggest that the strawberry aroma was in the dairy drink, but we actually took it out of the dairy drink and we supplied the panelists with the aroma using our olfactometer. In one kind of regime, we took the native aroma release profile as if the aroma was in the dairy drink, but we compared it to a situation where the aroma is much more prolonged in terms of its release. Now, this is a bit exaggerated. This is typically the release pattern you get from eating a block of cheese or eating a gummy bear of any kind of sweet, and you typically see the mastication behavior in a very prolonged release. What we actually took is an aroma release profile which was 4 times the aroma release, I would say, of the native aroma as it would have come from the dairy drink. So still we have a short aroma release profile as if the aroma was from the dairy drink, and a much longer aroma release profile as if the product was much more firm or was much more dense. And we asked these people, please judge yourself the rate of satiation you experience. And again, people got exactly the same amount of the dairy drink, hence they received exactly the same amount of stomach filling and the same amount of calories. But the basic question, please rate your level of satiation. And the level of satiation they were measuring, it has nothing to do with stomach filling, it has nothing to do with the calorie intake, it was just based on the sensory percept and the judgment in how that translates to, I would say, calorie intake. So what we've been doing is we've been monitoring satiation, the sensory satiation that is, versus time, depending on a prolonged aroma release profile, which we know that coincides with any kind of product which is high in fat and high in sugar. And we compare the situation to a product with a very short aroma release pattern, which is very much associated with a low fat product and a low sugar product. And then when you do this kind of experiment, you see actually that the prolonged aroma release profile provides much more sensory satiation to a person than a short aroma release pattern. And now it's starting actually to make sense. You can imagine that people subconsciously measure the timely release of the aroma and actually translate this into kind of sensory percept in terms of its levels of satiation. Now, as I mentioned, or in fact I stressed, this satiation is not really, has nothing to do with stomach filling. It's the sensory concept, it's the sensory percept of being satiated. And you can easily change this level of satiation, or you can easily change the attribute satiation by, for instance, indulgence or reward or happiness. So the basic question could be, for instance, how happy do you get from eating this foodstuff? And that makes it an extremely interesting study. So the model that we've been building is to translate this, in fact, to I would say, the emotional state of mind that people perceive from eating food. We also know that this prolonged aroma release profile is very much associated, for instance, with all flavors. Most of the all flavors are very lingering, very persistent in their perception. This also could tie in, for instance, the role of some of the artificial sweeteners, which have a very much licorice, or some of them at least have a very strong licorice aftertaste. And so you can well imagine that, and I'm not saying this is a conscious process, because many of the flavor experiences are being subconsciously processed in your brain, that people build themselves some kind of conceptual idea of the amount of calories, and probably also from the quality of food, not only by the flavor quality, in terms of the exact composition of the aroma, but also by means of the aroma release. It also starts to explain, in fact, why people one of the explanations, at least, is that people drink much more beer and colas than they drink milk. If you would drink milk and look at your tongue, you're going to find it covered with proteins, and you're going to find it covered with fat, which typically gives you a long aroma release profile. While something like any kind of cola or I would say beverage, soft drink, comes up with a very spiked aroma release profile. Most of them are pretty acidic. That means that your saliva production kicks in rather heavily, which is in a very effective way of removing this kind of beverages from your oral cavity, and that actually provides you with a very spiked aroma release profile. Now, as we've been showing, this is a non-satiating condition, this is a much more satiating condition, and it could easily translate in the fact that creamy products, typically creamy products, are being consumed in typically smaller volumes than anything which is non-creamy. So what you can learn from this exercise is that moods are very much affected by the way you perceive your aroma system. And now I'm going to switch back to Harold, because he also shows that mood can be a property of many intrinsic properties of the machines or the equipment of foodstuffs that you are producing and developing. So Harold, the floor is yours again.
Hello again. The part that Peter has been presenting is entirely on how to present something to a person. So you can go quite far in mimicking products to a person and present them to a person, and then measure a response to those. And the part of measuring the response is what I will continue on right now. It's not just the sweetness or the texture aspects or the lingering of the aftertaste that we could be asking for, that we could be interested in, but actually the real questions often are on will a person really like this food, or will he feel pleasant after eating the food, or maybe the opposite. And those questions are very relevant, and you can ask them in consumer research. What we would like to do is we would like to ask them also in the context that Peter just described. So if you have people sitting in a chair and all kinds of odor, taste, texture combinations are presented, and even environmental odors are manipulated, for instance, then we would like to know also there, what does that to a person? For that, you can ask a lot of questions to a person, but preferably you do actually a combination of asking questions and measuring things that are implicit measures of mood. And implicit measures of mood are those aspects that of behavior that people show, but that they actually do not consciously have to write down. So you can pick up facial expressions maybe, and other things, and that's something that we used in this study. We had a study on instruments that were prepared by Philips. This was presented in the conference once before, so I will be repeating that. Maybe some of you have seen this information, but the Philips Senseo coffee maker was used to add several aspects to it that could make it more fancy, and those aspects were the production of steam during coffee making, the illumination of the cup, the production of sound during coffee making, and also the production of aroma during coffee making, so therefore a coffee aroma, of course. The question was, does that do anything with the mood of the people? And as many of you know, drinking coffee or getting prepared to drink coffee already sets you in a certain mood. People anticipate with pleasure the drinking of coffee often, and all these aspects that you see here, aroma production and also steam and cup illumination, all have an effect on that anticipation of drinking the coffee. That's at least the way that we started doing this work. And how do you measure that mood? That's now the question. And in this case, we use a combination of aspects of people's responses, people's behavior. And also the environment was very well controlled for doing that. We used what you can call mood rooms, in which people can sit on a table. This is a table. You see a normal display of sugar and cups and stuff that you need for coffee drinking. This is the coffee maker. So people will be taking place here, and once they do, then the coffee maker is, for this picture, on this side. So they can all watch the coffee maker that is used to make their coffee. We make coffee for them. we make sure that during the coffee making people have nothing else to do, and since it is a very stimulus low environment, they do pay attention to the coffee maker, although we don't tell them, of course, that that is what the important part of the study is. These people were actually thinking that the coffee itself was the product that would do something with their responses. They were not aware of the fact that we were curious for their responses to the effects of the maker. What we did measure, though, in the time that people were here, these people are wearing heart rate monitors. And heart rates are continuously collected during the experiment. And as you see, people are also filling in forms, and these forms are really quick questionnaires with questions on different aspects of mood that they could fill in. It's an existing questionnaire that is used for mood inventories. So it has been validated also for larger populations. The odor in the room was also controlled as a function of the time of the presentation of the preparation of the coffee. And I will explain you something first on the heart rate data that we collected. So this is a typical electrophysiological response that you measure when people's hearts beats are picked up with an electrode. And over time that could look something like this. So this is over a period of 50 minutes, and you see that initially the heart rate is a bit higher, the frequency, and then it goes down. And this is actually the frequency of the interbeat intervals of the heart rate. And you see that after a while it goes up again. So this is a typical graph that you see when people sit in a chair, do nothing else than just write down some things and watch and talk. So there's no exercise in this case. During this stage you see that heart rate goes down a bit because people were actually walking before we started. So this is the rest period. You can define different sections during that time that are related to the conditions of the experiment. So here people are sitting, they receive instructions, and they don't do anything else. This is a resting period still. There are no task instructions, but we use measurement from this period as a baseline. And then after that we do start doing things for the coffee maker, and people start drinking coffee, for instance, and there are two moments where we take measures, and one is actually during the coffee making, and one is during the drinking of the coffee. If you focus in on that, this is what you see. Heart rate goes up and down, and what is peculiar here is that you see that there is a rhythm of heart rate going up and down, and that rhythm has a specific meaning, which I will explain to you now. These rhythms, you can reflect them as something related to frequencies going up and down, and they are linked to the respiration frequency of a person. So if you are relaxed and you do breathe in, breathe out, then you do see that under the condition that you are relaxed, that heart rate goes up and down in the same frequency as you're breathing. The less relaxed you are, the less you will see that this happens. So if a person is very stressed, you will often see a very constant heart rate without much fluctuation. So therefore, the fluctuation in itself of that heart rate is used as an indicator for relaxation. And you have different measures, and I've been looking at all of them. So these are all different ways to represent fluctuation. You can use standard deviation, you can use a frequency-based ratio, or you can use only the high frequency, which is actually the breathing frequency in this case. And condition is that people are not physically active. Now, the results of these measures for the experiment are that if you have the different conditions that you use, which is the basic product, the steam enhanced product, the lighting conditioned product, or the sounds product, then you see actually that for each one of them during coffee drinking, so this is coffee drinking, you will see that heart rate variation goes down with those bells and whistles. So the most fluctuation you see in the heart rate is for the basic product, and not for those that have different aspects. What is also important to know is that this is done during drinking, although the coffee machine was not working at that point in time. So people drink coffee, and then you see these effects, although they were introduced already previously when the people were looking at the machine when the coffee was made. And what is more interesting is that those same effects were not seen during coffee making. So people see something, and then the effect of that is only seen in their heart rate response during the drinking. We think we can explain this because actually the drinking itself is the moment where you ingest the product that you have seen being prepared before. And if there's something different in the preparation that could actually upset you, and most of that upset mood you might experience during the drinking, when it's the moment there to drink what you saw being prepared. So this is what we think has happened. These people were not used to steam and light and sound, and they trusted maybe the product a little bit less. So that was also the outcome of this product, of this experiment, that people were, for new machines, they were a little bit less relaxed when they had to drink the coffee. As you saw, people were also filling out mood questionnaires, and that is important still to us because the measures that you see on heart rate do not really say anything on the specific mood, only on the amount of relaxation. So we would like to know also whether the mood itself has a specific quality. And we used the US University of Wisconsin questionnaire, and they can be filled out quickly, and the scores result in something that has 3 values. One has to do with the hedonic state of the person. Do you feel fine? Something with the tension state. Do you feel tense or relaxed? And something with the energy state. Do you feel energized or do you feel tired? So those 3 aspects are extracted from scores on a list of attributes that people have to fill out in that form. We use only a selection of these actually. You can use them for a large number of settings. Effects of medications are studied, environmental factors, and also the effects of food preparation, like you see here. And for this experiment, the interesting thing was that for the mood, the most important factor was not one of those 3 that I showed you before, but was actually the odor that was released in the room. So what you see here is that this is tension scores, or the differential tension scores with respect to baseline. And you see that people are much more relaxed, less tense when the odor is presented during coffee making. And this is the case where there was no odor during coffee making. So the release of odor was a very important aspect. The more pronounced the coffee odor is, apparently, during coffee making, the more relaxed, the less tense you are during drinking. And that was most observed for the basic system, but even still for the systems with extra functionality. So you see now that the combination of those 2 aspects, which is the heart rate, physiological response, and this quick questionnaire, gives you a very complete view of the way people feel during a task, during eating or drinking a product. And the last thing here is that much of that is measured without the people even realizing it, and you can still use it in a natural environment like the coffee drinking environment that you saw in the pictures. This is just the start of what we are doing right now and what we will be doing in the future, which is a project in this case where we are working with European funding in a project that has as a final objective to deliver a system that we call the food cognition simulator. It's a system that can simulate visual, auditory, taste and texture and sound and aroma aspects of a product, and also of the consumption environment of a product. And the environment can be something that has to do with the advertising that you use, or the packaging, or the way you place it in the supermarket. All that can be mimicked. A person can even go into the supermarket and virtually and can be exposed to all that information, and we measure a number of responses to that environment, and even also the taste, texture and caloric inputs can be controlled during experiments. And those output factors have something to do with the focusing of the eyes on different aspects of the environment, measures of blood flow to the brain, which we don't do by MRI actually. We do that with a system that uses infrared light detectors on the scalp to pick up blood flows really underneath the cranium actually. And we use EEG and we use heart rate, that's like I showed you before. Critical here is that you need actually something to interpret all these responses, and that's the major effort of this project now, that we do pick up all these responses to different kinds of situations, and we also know what people will do in fact when they would have to have the choice of different options of products. So we use that information also to pick from all the measures here the best predictors for their behavior. And that is what we will be doing in this project, and what we will deliver hopefully in 2 years from now. And even aspects like buying intent is something that we would like to to be able to predict better than we do now. FOCOM is the project, so it's full cognition modeling. It's the abbreviation of that. It's sponsored by the European community, and we are running it in the Netherlands. So that was the talk. We have been running a bit late in time. I hope that everybody still had time to follow it until the end. Sorry for the interruption actually we had in the middle, something you cannot predict, of course. But hopefully for you it was not something that stopped you from enjoying it. So just to close down, I would like to invite you, if you have any further questions, like there were several that we couldn't answer immediately, we put it up with you. If you have questions that you have not stated yet, we will be happy actually still to receive them, and we will deal with it in a later stage. So again, thank you very much for paying attention and for your interest in the subjects that we had today. And we hope to see you back next time in the future for follow-up courses that we will have also on different subjects. Thank you very much and goodbye.